exploring the mitotic division of an animal cell.
The first step is G2 of interphase. A nuclear envelope surrounds the nucleus. Each centrosome features 2 centrioles. Chromosomes cannot be seen individually because they have not yet been condensed.
The first actual step of mitosis is prophase. In prophase, chromatin fibers become more tightly coiled, condensing into discrete chromosomes. The nucleolus disappears. Each duplicated chromosome appears as identical sister chromatids, joined together at their centromeres, and along their arms by cohesion. The mitotic spindle begins to form composed of centrosomes and microtubules. The centrosomes begin to move away from each other due to the lengthening of their spindles.
Next is prometaphase. The nuclear envelope fragments. Chromosomes have been even more condensed. Microtubules extend from each centrosome into the nuclear area. Each of the 2 chromatids of each chromosome now has a kinetochore.
Metaphase is next in the process of mitosis. Metaphase is the longest stage of mitosis. It can last about 20 minutes. The centrosomes are now at opposite poles of the cell. Chromosomes line up on the metaphase plate. The chromosome's centrosomes lie on the metaphase plate. Each chromosome's kinetochores are attached to the microtubules coming from the opposite poles.
Anaphase is the shortest stage of mitosis. Anaphase begins when the proteins are cleaved. This allows the 2 sister chromatids of each pair to part suddenly. Each chromatid now becomes a chromosome. The 2 daughter chromosomes begin moving towards opposite ends of the cell as their kinetochore microtubules shorten. By the end of anaphase, the 2 ends of the cells have an equivalent and complete collection of chromosomes.
The final step of mitosis is telophase. In telophase, 2 daughter nuclei form in the cell. The nuclear envelopes begin to form from the fragments given from the parent cells. The nucleolus also reappears. Chromosomes become less condensed. Mitosis is now complete. It has successfully created 2 genetically identical nuclei from 1 nucleus. Cytokinesis happens after mitosis is complete. The cytoplasm of the cell successfully divides during cytokinesis. In animal cells, a cleavage furrow forms and pinches the cell in 2. In prophase 1, chromosomes begin to condense. Homologs are loosely paired along their lengths, aligned gene by gene. Crossing over occurs while homologs are in synapsis. Chiasmata form where crossing over occurred. Centrosomes begin to move, spindles begin to form, and the nuclear envelope breaks down. Each homolog is attached to kinetochore microtubules. Homologous pairs of chromosomes line up on the metaphase plate.
During anaphase 1, each pair of homologous chromosomes separate. The homologs move towards opposite poles, guided by the spindle apparatus.
2 haploid cells form during telophase 1. During cytokinesis, the cytoplasm of each cell completely divides. During prophase 2, the spindle apparatus forms. Chromosomes are still composed of 2 chromatids. Metaphase 2 is almost the same as in mitosis, but however, because of crossing over done in meiosis 1, the 2 sister chromatids are not genetically identical. Kinetochores from sister chromatids are attached to microtubules extending from opposite poles of the cell. In anaphase 2, proteins holding sister chromatids together at their centromere breaks down, allowing the chromatids to separate. The chromatids move toward opposite poles as individual chromosomes. In telophase 2 and cytokinesis, nuclei form, the chromosomes begin decondensing, and cytokinesis occurs. The meiotic division of 1 parent cell produces 4 daughter cells, each with a haploid set of unreplicated chromosomes.
The importance of mitosis is cell replication. One of the differences between mitosis and meiosis is that mitosis has 1 cell division, while meiosis has 2. Mitosis starts out with 3 diploid parent chromosomes and produces 3 diploid daughter chromosomes. Meiosis starts out with 3 diploid chromosomes and ends up with 3 haploid. Meiosis 1 separates homologous chromosomes, while meiosis 2 separates sister chromatids. This concludes our exploration of cell division.